Hello everyone, today we are going to learn about sexual reproduction in flowering plants, in which we are going to first start with the male reproductive organ, that is androecium. Androecium, which is also known as stamen, is divided into two parts. It has the anther and filament. Anther, which is connected to the filament with the help of connective. Now if we look into the structure of anther, if we cut a transverse section of anther, we will find that to the outermost part there are four layers. The four layers are, starting from the outer part, it is epidermis, followed by endothecium, then comes middle layer, and the innermost is tapetum. The outer three layers, they are associated with the function of providing protection, and not only this, it also helps in the dehiscence of the anther, which further helps in releasing the pollen grains when they are mature. Then comes the tapetum. Tapetum has an important role to play. The tapetum is the innermost layer surrounding the pollen sac, and it helps in providing nourishment to the developing pollen grain. Next, within the pollen sac, you can see several cells present. These cells are called as porogenous cells. The term genus stands for production. What do they produce? They produce spores, which we call as microspores in case of the male part. So microspores are being produced by these porogenous cells. Before that, they undergo mitotic cell division, resulting into the formation of PMC, which stands for pollen mother cell, which may also be called as microspore mother cell. This is the cell which is about to undergo meiotic cell division and would further form the pollen grains. Each pollen mother cell produces 4 pollen grains, each of which is a haploid cell. Now, starting with microsporogenesis, which means production of microspore. Microspores are been obtained from PMC, that is pollen mother cell. It undergoes meiosis. Meiosis occurs in two stages. So as the meiosis 1 occurs, it gives rise to a 2-cell stage, which we may also call as the microspore dyad. Further, it undergoes the second stage, forming the microspore tetrad. As this undergoes further development, it releases the 4 pollen grains. All these pollen grains get detached. Each microspore, which is now a mature pollen grain, is ready to be released. A pollen grain that you see here would be having 2 layers. The outer layer is called as exine, inner layer is called as intine. Exine, which is composed of a very important bioresistant material known as sporopollenin. It helps in making the pollen grain resistant towards extreme conditions. It can be strong alkaline condition or strong acidic condition, towards high temperature as well as towards extremely low temperatures, making it a good fossil. The inner layer that you see is called as intine. The intine is composed of 2 chemicals. One is cellulose and the second is pectin. Cellulose and pectin together form the intine. There are regions where there is absolutely no exine present, which is called as the germ pore. It is through this germ pore that the pollen tube arises during pollen germination. Here there are 2 cells present, the larger one and the smaller one. Larger one, which is named as the vegetative cell, and the smaller one is called as generative cell. Thank you.